everyone. My name is Manus Boy. This is State 2 AP Biology. Today I'm going to be talking about cell signaling. And quick context, we know that our bodies have millions and billions of cells and they all communicate with each other, just like how you and I, we all communicate with each other too. Alright, but seriously, without cells communication, we wouldn't know, cells wouldn't know when to divide, when to repair, or when to heal, or how to react or interact with the environment. In short, basically, cells run everything and keep everything in sync. I like to kind of think of a cell signal and receptor as a kind of a lock and key relationship. Signal molecule is the key and the receptor is like the lock. Only the right signal fits in the right receptor, so the cells don't get mixed signals. Depending on how far a signal needs to travel, cells use different ways to communicate. So some travel short distances while others travel long distances, and some just move from neighboring cells to other neighboring cells. Let's start off with the endocrine signaling, which happens over long distances. Here we have special hormones released into the bloodstream. These hormones travel through the bloodstream until they find their matching target cells with matching receptors. For example, in real life, the hormone insulin made by the pancreas travels through the blood to help regulate blood sugar levels. And another example of this is the adrenaline, which spreads through the body, which help us prepare for fight or flight, like as a response stimulus. Because of this, endocrine signaling kind of reaches far, but it takes time. It's slow because the signals move through the bloodstream, which moves throughout all parts of the body, which can take time before it can respond back. We have the autocrine signaling, auto meaning self, and this is when a cell sends signals all the way back to itself. This is mainly for cell regulation for its own behavior. For example, certain immune cells uses like signals to activate themselves or strengthen their response in a way. In fact, some release growth factors that signal themselves to grow and divide, helping tissues heal or replace damaged cells very easily. In my own personal opinion, I like to think autocrine signal as a cell giving itself a reminder or instructions to act on its own. So here we have the little thingy moving from the signal molecules to its receptors, which kind of binds to itself right here or here. So next we have the pancreas signaling, which happens over short distances. So it's technically just neighbor to neighbor cells, from regular cell to its target cell. So it's just neighbor, hi, how are you? I'm doing good, sort of thing. So here the signal molecules go from signal 1 to the target receptors of the target cell. And a real good example is when in a nerve cell, nerve cells release neurotransmitters, which passes message from basically one nearby cell to the next. And because of this, we know that signaling molecules and pancreas only affects nearby cells in a nearby area. Another short distance signaling type is the juxtacrine signaling, which is all about direct contact with another cell. Here, signaling molecules kind of stay on one cell surface and connects with a receptor of another cell surface. It's like two cells basically shaking hands and passing on the next message to one another. This, however, is super important because cells need to coordinate what they're becoming and where do they go. And this whole metabolic process is what shows that cell development, it goes hand in hand with cell communication. But not least, we have the gap junction, which unlike the short distance like juxtacrine or autocrine or paracrine, that direct contact is between two cells with a protein channel. Most of these proteins are called connexins. When 6 of them come together, they make like a full connexin, which is like a half tunnel. And when two half tunnels bind together, they make a full bridge. So here these yellow lines are representing the connexin. And this is where signal molecules travel through from one cell to the target cell. The most important part about this is that these tunnels allow small molecules and ions to pass through from one another, which helps processing things much more easily. Perfect example of this is like in the heart. Gap junctions let electrical signals move smoothly between heart muscles, which keep your heart kind of have a nice and steady beat. I said last but not least for the gap junction, but it's because quorum sensing is between prokaryotes and other prokaryotes. Or a prime example would be bacteria. Generally, in quorum sensing, bacteria kind of like releases small molecules to sense like how many of them are nearby, and once enough signals are built up, meaning the population is dense or have like a higher concentration, all of these bacteria work together as a group. A little example I could give is that one bacteria, the Vibrio viscerea bacteria, uses quorum sensing to make the Hawaiian bobtail squid kind of glow in the dark. But this only glows in the dark when there's enough bacteria to make light noticeable or to make visible light. Technically, that isn't the only example I could give, because there's other bacteria such as like the Pseudonoma aeruginosa, which uses biofilm formation. You're probably asking, what is biofilm in general? Biofilm is technically a protective layer of bacteria that helps bacteria like stick to surfaces, and yeah, that's pretty much it. And because of this, quorum sensing helps these bacteria such as the aeruginosa decides when to form biofilm and when to release toxins after reaching a certain population or density. That's specifically what actually kind of makes them harder to kill with antibiotics, which is kind of why biofilms are a big problem in medicine right now.